To initiate a transgenic mouse genotyping service request with UAGC, make sure you're logged in to the iLab operations software. Once you're logged in and at our course landing page, please click the tab that says Request Services. This will pull up a list of available service request types. Click Transgenic Mouse Genotyping to expand this category, and on the right, click Request Service. This will open up the Transgenic Mouse Genotyping request form. This is the table where you will enter your sample barcodes and the corresponding assays to be tested for each sample. It's probably easiest to download the template and fill this out on your computer, and we'll upload it to the form here. Click on the TGM submission template. This will ask that you open it up in Microsoft Excel. Here is the template open in Microsoft Excel. First, you'll enter your sample barcodes. These must be the barcodes that are on the tubes supplied by UAGC. If you have not received transgenic mouse genotyping sample collection tubes from our lab, please contact our lab to obtain these tubes prior to submitting your batch. I have entered my barcodes here. This column, Animal ID, will not be used by the UAGC as part of the testing or reporting process. Some labs may find this column helpful, so that way you may track and record which of your animals the UAGC barcode applies to. Please keep in mind, UAGC will only report results in terms of the U of A sample barcode found on the tubes, so please be sure to carefully record which animals these barcodes correspond to in your facility. Next, we're going to order assays for each sample. Any assays ordered on the same row of the spreadsheet with the sample ID will be used to test on this sample. These fields, TGM assay 1, 2, 3, 4, and 5, allow you to pick from our most common assay list. Select the cell here and choose from an assay on the list. To apply this assay to more than one sample, you have two options. First, select this cell with the known assay in it. Select all other cells to apply this assay to, and on a PC keyboard, click Ctrl-D. This will translate beta 2 to all of these cells. To order more than one assay per sample, please click another column. Again, select from our drop-down list of our most common assays here. Another option to get this information into the following cells without changing it is to hover in the corner, generate the cross cursor, hold down Control, and you'll see your cursor becomes a cross with a small cross on the upper right. Now, hold and drag down while holding the Control key to ensure this assay is translated down the row. You do not need to select more than one assay or region of interest per sample, but many labs do find this necessary for their protocols. You can select up to 5 assays per sample. Again, these columns will only accept assays from our most common list. If you enter an assay that is not on our most common list, an error will occur. To order assays that are not on our most common drop-down list, but are found on our pre-validated assay list, enter them here in the TGM Other Assay List column. Different assays must be separated by a comma. Finally, we want to make sure that all samples have at least one assay ordered for them. I'm going to select this assay and apply it to the rest of my samples. Ctrl-D will fill this in. Once we are satisfied that each sample has received an order for all of the requested assays, we're going to copy this information and paste it back into the iLab submission form. Do not copy the column headers, and it's not necessary to copy any empty rows. Select all rows that contain a sample barcode, copy on your computer, and go back to the iLabs form. Once you're back at the iLabs form, let's paste in this information to fill out the sample table. We'll select all rows across the top and paste in. You can scroll through this table here to see that your samples have maintained the assays that you intended to order. Once we're satisfied with our sample list and the assays ordered, we'll proceed through the form. you'll be asked to enter the total number of sample tubes submitted. In this list, we have 16 distinct sample tubes. Click Add Selected Services. Next, you'll be asked to enter the total number of assays requested. Please be sure to carefully count how many assays you've entered, counting duplicates per sample, and don't forget, over here, each assay is separated by a comma. I've selected 33 distinct tests. Again, click Add Selected Services. Once we've added the proper number of sample tubes and the number of assays, we can see down below here, iLabs is starting to generate a cost estimate for this project. Please proceed to complete the following required fields in this form, including reading and acknowledging the end of project terms statement. You'll be asked to agree to the same statement each time you submit an order to our lab. Once you've completed all required fields, click Save Completed Form. Next, you'll be asked to enter payment information. If you're a U of A internal user, this will be a drop-down of your available U of A accounts. If you're an external user, please enter your PO number here, or if you prefer to pay by check or credit card, enter that information here. Finally, we're going to click Submit Request to Core. This is your request dashboard, and this is the submission we just created. It is very important that you copy down the numbers at the end of your service ID and transcribe these numbers to your physical submission. This may be transcribed onto the container of your samples or baggie that your tubes are in, the box or shipment, and must be attached to your physical submission in order for our laboratory to correctly identify your samples when they arrive. Your results and any communications specific to this submission will take place and be posted to this submission in the iLabs platform. Please contact our laboratory if you have difficulty submitting or questions about this process. You can email our lab at uagc@email.arizona.edu. We look forward to working with you.